We are doing Unit 4, Topics 6 and 7. This is the cell cycle. You're going to take notes starting on page 35, and we're going to go to the end of the unit. So fantastic. Let's get started. So, cell cycle. This is a process in which one cell becomes two, right? So it's a cell division process. It's integral to life, right? Without this, we wouldn't be multicellular organisms. This allows for the reproduction of cells, the growth of cells and tissue repair. Without it, our cuts would never heal. The cell cycle, the life of a cell from its formation until it divides. That's sort of just the definition of it. So before we get into cell cycle, let's talk a little bit about how DNA is organized inside the nucleus. So cells must organize and package their DNA before division. DNA associates and wraps around proteins known as histones. So these are kind of like, a lot of people describe this as beads on a string, right? The histones are the beads and the DNA is the string, and it kind of wraps it around. This forms what we call nucleosomes. So histones and nucleotides are nucleosomes. A string of nucleosomes. This forms what we call chromatin. So again, we have stranded DNA. It wraps around histones to form nucleosomes. Strings of nucleosomes are called chromatin. When a cell is not actively dividing, chromatin is in its non-condensed form. So we don't have to condense it. It's just kind of hanging out willy-nilly when a cell is not actively dividing. After DNA replication, which we're actually going to get into in next chapter or next unit, chromatin condenses and we have what we call a chromosome. So chromosomes are what you can see under a microscope. It's a more familiar term to you. That's what occurs when chromatin condenses. Chromosomes are densely packed. This allows for easier division. It also allows for, and again, we're going to get into this in the next unit, it allows for regulation of gene expression as well. So if a chromosome is densely packed in one area, it can't get transcribed. Great. So here's what it kind of looks like, right? Since DNA was replicated, each chromosome has a duplicate copy. So you got one here and one there. We call those sister chromatids. They are replicas of each other. Copies joined together, they form, as I just mentioned, sister chromatids. So each one of these strands of DNA, each one of these chromatids is an exact copy of the other. We have this centromeric region, or this centromere. It's the region on the sister chromatids where they're closely attached. It's a region. It's not a thing, right? There's not like a little thing there. It's just a region of those sister chromatids. It's like where they're holding hands. Microtubules attach here. We call it the kinetochore. There you go, kinetochore. Proteins attached to the centromere that link each sister chromatid to the mitotic spindle. So like they're little hooks that the mitotic spindle comes and attaches to before they pull them apart. Here's everything, right? So we have DNA in its just natural form, right? We condense it around histones. These histones form nucleosomes. Nucleosomes, again, they look like beads on a string, right? So the string is the DNA, and then the beads are the histones. When that wraps and wraps and wraps and wraps, then we have chromatin. That chromatin condenses even further, and we have a chromosome. When chromosomes pair up, they're called sister chromatids. And again, they're all hanging out in the nucleus. So this is how we get 8 feet of linear DNA 46 times over into a nucleus smaller than a cell, is that we just wrap it. Great. So genomes. A genome is all of a cell's genetic information. So it's all the DNA. In prokaryotes, it's singular circular DNA. You guys know this from Unit 2. In eukaryotes, it's one or more linear chromosomes. Humans have 23 pairs. We have 46 chromosomes. Every eukaryote has a specific number of chromosomes. So that's for humans, right? Humans have 46, but chimps have 48. Elephants have 56, right? The one thing I can tell you is that it's always even numbers because we are paired up. Chromosomes are paired up. So you can kind of see over here, there's a gene here and a gene here. The whole thing together is a chromosome, and then get all those chromosomes. This is your genome. This is everything. Okay, homologous chromosomes are two chromosomes, one from mom and one from dad, that are the same length. They have the same centromere position. They carry the same genes, controlling the same characteristics. So, a lot of biology textbooks and that sort of thing like to, differentiate them with colors. One's from mom, one's from dad. When we talk about they carry the same genes, different genes are located on different parts of your DNA. So, for example, the gene that codes for eye color, right? That's going to be matched up with another chromosome over here that also has the gene for eye color, right? So they're controlling the same characteristics of the organism. When they replicate, here we go. Now we just have the homologous chromosome 1 and homologous chromosome 2. These are your sister chromatids when they match up. So this one right here is an exact copy of this one right here. This one right here is an exact copy of this one right here. And they come from mom and dad. Cool. Practice. Free response question. Biological structures tend to be composed of smaller units that assemble into more complex structures. Using eukaryotic chromosomes as an example, describe the smaller units in their assembly that leads to a larger, more complex structure of a chromosome. Identify one major function of chromosomes. I'm sure you're going to say something like, oh yeah, just like macromolecules are comprised of smaller subunits, DNA is also comprised of really small units of nucleic acids that are then assembled together, that then form a line of nucleic acids that wraps around histones, that then condenses into chromatin, and that condenses even further. You see that? You probably said something like that, I'm sure. Identify one major function of chromosomes. Well, it carries genetic information. Derp. All right. So when we're talking about mitosis, and next unit we're going to get into meiosis, we're talking about two types of cells. So you have somatic cells. These are your body cells. We call them diploid because it has two sets of chromosomes, one set from each parent. Right? These are your body cells, your skin cells, your eye cells, your hair cells, your intestinal cells. They divide by mitosis. In humans, we call it 2n equals 46 because, again, it's two sets of chromosomes. Our chromosomes pair up. 23 from mom, 23 from dad. Love it. Gametes are the eggs and sperms, right? We call those haploid. That means they have one set of chromosomes. They divide by meiosis. Again, we're going to look at that in the next unit. In humans, n equals 23. This unit, we're going to focus on mitosis, right? But you may notice something right here is that gametes here are eggs and sperm, and they have 23 chromosomes each. And if you take 23 and times it by 2, we got 46. So half from mom, half from dad. Okay, here's the cell cycle in all of its glory. It consists of alternating phases of interphase and mitosis. We're going to get into interphase in just a second. We have G1, the G stands for gap. We have S stands for synthesis. We have G2, again, G stands for gap, and then mitosis, and then cytokinesis. I want you to note too that in this entire cell cycle, this is the only portion that the cell's actively dividing. Through the rest of it, it's growing, it's synthesizing a new strand of DNA, it's double checking to make sure that there's no mistakes, that kind of thing. So it's a little bit more complicated than you may have learned in other classes. So the G1S and G2, we call that interphase. Again, it's when the cell is growing and getting more stuff and replicating its DNA. Mitosis is the actual splitting of the cell, and then cytokinesis is the splitting of like the cytoplasm. Okay, let's talk about interphase. It's the longest portion of the cell cycle. The vast majority of cells are in interphase. If you look at them under a microscope, about 90% of them are just kind of hanging out, waiting to divide. G1, we call that the first gap phase. In that phase, the cell grows and carries out normal functions. Remember, if we're going to make two of something, if we have one and we're going to make two of something, we got to double all the stuff inside. So in the first gap phase here, the cell is doubling the cytoplasm. It's making more lysosomes. It's doubling the amount of mitochondria, right? It's preparing to split. S phase is the synthesis phase. This is when DNA actually replicates. We're going to get into this again in the next unit. It's a fascinating process. So DNA will replicate and chromosome duplication occurs. So again, we're trying to make 2 of everything. The G2, we call that the second gap phase. This is the final growth and preparation for mitosis. There's a couple checkpoints in here too. We're not going to duplicate a cell that's not ready to be duplicated. So this is also where a second check happens. We'll get into that at the end of this lecture video. And then you have the M phase, which is actually mitosis, right? This is when the nucleus divides. Cytokinesis is when the cytoplasm divides. So they're 2 separate steps. First, the nucleus divides, then the cytoplasm divides. That's why we kind of break those 2 things up. Mitosis then results in 2 identical diploid daughter cells. That's 2N. Diploid daughter cells, 46 chromosomes each. It's a somatic cell, not a gametic cell. So it's your body cell. So here it is in all of its glory. We have phases, 5 of them: prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. Telophase and cytokinesis is kind of broken up to, is kind of, grouped up together. And then there's this fun new phase that has been introduced since I took this class a million years ago called prometaphase. Okay, let's go over each one. In prophase, there are some key events that happen. First, the chromatin condenses. The nucleoli disappears. So if you take a look at a cell inside the nucleus, you'll see like a really dark kind of dot. That's called the nucleoli. It is a really kind of condensed spot of DNA. So you're going to see that disappear. The nucleus is going to kind of get hazy if you're looking at it under a microscope, which we will do. Duplicated chromosomes appear as sister chromatids. So you'll start to see sort of like the X shape looking thing. Mitotic spindle begins to form. It just starts to form. We start to see it form from the centrioles. And then centrosomes move away from each other. That's these things right over here. They build the mitotic spindle. Centrosomes, centrioles, sort of for the purposes of this class, the same thing. Okay, prometaphase. So these are 1, 2, 3, 4, 5 key events that happen in prophase. Prometaphase, key events that happen. The nuclear envelope fragments, right? So we got no more nuclear envelope. It breaks apart. Microtubules from the centrosomes appear, attach onto those kinetochores, those regions on the chromosomes, on the sister chromatids. They attach to, and that's it. That's what happens in prophase. I'm sorry, prometaphase. 2 key events. In metaphase, this is the easiest one to recognize under a microscope. The centrosomes are at opposite poles. So we got one over here and one over there. So they kind of move to opposite sides. Chromosomes will line up across the metaphyseal plate or the metaphase plate. It's just a region in the cell where they line up. It's not actually a plate or anything like that. Microtubules attach at each kinetochore. You can kind of see that in this picture here. So we got 2 attached to this one chromosome. And that's it. 1, 2, 3. 1, 2, 3 key features in metaphase. So you'll start to see sort of the polarization of these centrosomes and the lining up of the chromosomes. Anaphase, this is another really distinct phase that you can see under a microscope. So we have sister chromatids. They separate to the opposite ends of the cell due to the microtubule shortening. Okay, these guys don't move, these guys don't move, the microtubules shorten. That's really important to know because we will start talking about cancer and cancer treatment, and there are some individuals out there who believe that we can introduce drugs to this kind of process that prevents those microtubules from shortening. the cell elongates at that point too, right? So it kind of becomes this like egg shape. All right, 1-2 events in anaphase. Telophase and cytokinesis. There's some key events that happen here. So 2 daughter nuclei will form. So you'll start to see a nucleus kind of form on either side. Nucleoli reappear. So you'll start to see the dark circle inside of the nucleus again. Chromosomes become less condensed. Everyone kind of relaxes, takes a deep breath. So that would be telophase, right? Nucleus forms, nucleoli reappears, chromosomes relax just a little bit. 1, 2, 3 events. Cytokinesis occurs at this point. Animals tend to have this cleavage furrow that appears due to a contractile ring of actin filaments. So actin is a protein that assists in movement. Anything that moves inside the cell is helped by actin. Actin has another protein that it couples with called myosin. Very interesting stuff. Plants, vesicles produced by the Golgi travel to the middle of the cell and form a cell plate. Remember, there's a cell plate in between plant cells on the outside of their membrane. Okay, tracking chromosomes. So this is fun. Here's our parent cell. We have 2n equals 2. That means we got 2 chromosomes, right? So in normal somatic body cells, 2n equals 23, 46. 2n equals 40. Yes, 2n equals 46. Sorry, I had to like remind myself of that. So we have a parent cell here. 2n equals 2, so we got 2 chromosomes. One from mom, one from dad. Then each one replicates. So exact copy of the other. Here they are. They're going through mitosis, and now we have 2 identical diploid cells that are identical to the parent cell. Easy peasy. After S phase, 2 chromosomes and 4 chromatids. There we go. Anaphase, 4 chromosomes. 2n equals 2. Again, 2 chromosomes in the first one, 2 chromosomes in the second. So you could track chromosomes through each phase, right? So in prophase, and there will be questions about this on the exam. Prophase, we have 46 chromosomes. Metaphase, we have 46 chromosomes. Then all of a sudden, it doubles. Anaphase and telophase, and then finally, when they're separated again, you got 46. So at some point, it doubles in anaphase and in telophase, and then it separates once again. Great. Identify stages of mitosis in the image below. Oh, this one looks like interphase. You can see that because it's got that dark nucleolus. Prophase, that starts to disappear. I'm thinking this one right next to it is prophase. See how it's a little more hazy, the nucleus? Then we have prometa, which is probably a little bit like this. It looks similar to prophase. Nuclear envelope is breaking down, and some of those, you can see it's a little bit bigger. Metaphase, super easy to recognize. Anaphase, again, super easy to recognize. Telophase cytokinesis is a little bit tougher. This one up here is interesting. That's probably more of your prometa, right? It's sort of elongating. Anaphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Where's my cytokinesis? This looks a little cytokinesis-y. Okay. But you see how some of these phases are really easy to recognize? We'll be able to look at these under a microscope. Cool. All right. Now let's not do practice multiple choice. You guys can do that on your own. Okay, practice free response. A cell has 50 chromosomes. After mitosis and cell division, you find that one daughter cell only has 49 chromosomes and the other daughter cell has 51. Predict one way that this could have happened. Well, there's a couple ways that this could have happened, right? Maybe one of the microtubules didn't connect to the kinetochore very tightly, right? So one chromosome, instead of getting split, was sort of dragged onto one side, both of them together. It could have happened because of a replication error. It could have happened, there's a number of different ways. We'll get into chromosomal disorders, I believe, next unit. Yep. When we talk about meiosis. It's very interesting. Okay. It's a crazy process. How do we regulate it? We can't just like let it happen. That'd be wild. Cell cycle goes through checkpoints. These control points that regulate the cell cycle, and cells will receive stop and go signals. So if it's not, up to par, they'll say stop. We don't want you to divide anymore. We're going to skip this. What are some things that a cell might check during a checkpoint? Or if you want to turn and talk to, like, your pet or your parents, your sibling, or like a stuffed animal. Great. Let's talk about it. So at the G1 checkpoint, it is the most important checkpoint. You can see it right over here. Boom. It's long. It's the most important checkpoint. It checks to make sure that the cell has enough stuff, right? So it checks for cell size. It checks for growth factors, if there's anything that is missing that the cell needs in order to grow and divide. And more importantly, it checks for DNA damage. So there's a built-in mechanism in your body that if your DNA is damaged, we're not going to replicate that cell. Stop-go signals. So a go is cell completes the whole cell cycle, and a stop. So let's say you got a cell and the DNA is damaged because, I don't know, you like laying out in the sun and you get some UV rays. The cell will get a stop signal to stop dividing, and it goes into this, what we call G0 phase. This G0 phase is kind of like a waiting area. This is so the cell kind of has time to fix any of the mistakes that were made. If they can't fix it, the cell gets a signal to go through programmed cell death called apoptosis. So if it's not good enough, you're out of here. Okay, another major checkpoint. So G0, as I was just saying, some cells stay in G0 forever, right? Muscle cells, nerve cells. Your nerve cells don't grow and divide. They don't go through mitosis. They just grow longer. Your muscle cells, the same thing. They don't grow and divide very often. They grow in size. Some cells can be called back into the cell cycle, like if they get their stuff together and they fix their DNA, right? You can go back into the cell cycle as long as you pass that checkpoint. G2 checkpoint, right over here. So it's right after the DNA will duplicate. It checks for the completion of DNA replication and again for DNA damage. So we check for DNA damage over here before we replicate, and then we double check after we replicate. When we say go, DNA looks good, it's not damaged, it's replicated, we got 2 of each, cell proceeds into mitosis. If it receives a stop signal, the cell will stop and attempt to repair their damage, but if they can't repair it, the cell will undergo apoptosis, which again is programmed cell death. Boom, right there. Okay, let's talk about the M checkpoint, because there is a checkpoint right before mitosis. So this checks for right before the cells divide. It checks for microtubule attachment to the chromosomes of the kinetochore at metaphase. So it happens during mitosis, but before the cell splits. If everything looks good and the microtubules are attached to the kinetochore at the correct spots, then it completes anaphase and goes through mitosis, and now there's 2 of them. If a stop signal is reached because the microtubules didn't attach properly to the kinetochores, the cell will pause and allows for the spindles to finish attaching. So we like pause for a second. Okay. So those are all like major checkpoints and regulators in the cell cycle itself, but there's also internal regulators. So regulation of the cell cycle involves an internal control system. It consists of proteins known as cyclins. This is important too. The concentration of cyclins varies. They are synthesized and degraded at specific stages of the cell cycle. You're going to see this in a chart on the next slide, hopefully the next slide, and it'll be a little clearer. There's also enzymes known as cyclin-dependent kinases. So ASE, it's an enzyme, and it's dependent on this protein to exist. Concentration remains constant through each phase of the cell cycle. So talking about cell communication, there's sort of some in-talking that happens inside the cell in order to say, yeah, we're good, grow, divide, do whatever. CDK, cyclin-dependent kinases, are active only when a specific cyclin is present. That's where the dependent comes in. So cyclins come first, CDKs come second. So here it is. Sorry, these are think, turn, and talk. CDKs are active only when a particular cyclin protein is present. What would be the effect of a drug that prevents the degradation of the cyclin found in G2? The answer to that is effective drug that would prevent the degradation of the cyclin found at G2. The cell wouldn't go into G2. It wouldn't check for DNA damage after synthesis. If the cell can't fix it, then it would go through apoptosis. Okay, so look at this. This is cyclin expression cycle. Each cyclin CDK complex has a specific regulatory effect. Active CDK complexes phosphorylate target proteins, which help regulate key events. So check this out. We have a concentration on the y-axis, and we have different phases on the x-axis over here. You'll notice that cyclin D is pretty much active through the whole thing. That's sort of the cyclin that says, yay, we're dividing. After G1 phase, you see another cyclin peaks here and then decreases once we get into S phase. So this is kind of how the cell regulates whether it's dividing or not. Notice the varying levels of each of the cyclins at each stage. CDKs correspond to those cyclins. There's also external cell regulators. There are things called growth factors, right? These are hormones released by the cells that stimulate cell growth. It's a factor that stimulates growth. It's a growth factor. At this point, signal transduction is initiated. Remember G protein coupled receptors, signal transduction intracellularly. CDKs are activated, leading to a progression throughout the cell. So a signal from a growth factor is sent into the cell. A cascade occurs in which CDKs are activated. Also, contact inhibition or density inhibition. This is something that's interesting because this has to do with cancer and tumors. So cell surface receptors recognize contact with other cells, and it initiates the signal transduction pathway that stops the cell cycle. So you cut your finger, cells grow and divide, and then once they touch each other, there's a signal that's sent that says, stop, we're good, we don't need any more. Tumors don't have this contact inhibition. They lose this function, right? So grow and divide, when they touch each other, they just continue growing and dividing. And then anchorage dependence. So cells rely on attachment to other cells for an extracellular matrix to divide. So all of our cells are kind of attached to this extracellular sort of matrix, right? And without that attachment, we don't get the signal to divide or to stop dividing. Another factor. So all three of these, actually, really, if you disrupt any of these regulators within the cell, you get a tumor. So yeah, the genetics of cancer is fascinating. And it really has a lot to do with cell cycle, the regulators, and mitosis. Oh, speaking of cancer, so normal cells can become cancerous through DNA mutations. DNA mutations are changes in the DNA. We're going to get into this ad nauseum next unit. Cancer cells, on average, have accumulated 60 or more mutations on a gene that regulates cell growth. So all cancer really is a screw up of cell growth in the cell cycle. Too many cells, not enough regulators. So there are some differences between normal cells and cancer cells. Of course, normal cells follow checkpoints. They divide on average 20 to 50 times in culture, some less, some more. They go through apoptosis when significant errors occur. So if significant errors occur, the cell has a way to fix that by sort of removing itself from the equation. Cancer cells don't follow checkpoints. They divide infinitely, right? So think back to Henrietta Lacks with the immortal cell line. They divide infinitely in culture and considered to be immortal. They evade apoptosis and continue dividing even with errors. So you know, cancer is again a misstep in these regulators, these checkpoints. Yeah. So cancer cells, uncontrollable growth of a cancer cell can lead to a tumor. It's a mass of tissues formed by abnormal cells. Two different types of tumors. There's benign tumors. This means the cells are abnormal but not considered to be cancerous. Cells remain only at the tumor site and are unable to spread anywhere else in the body. So again, this is just an overgrowth of cells. A malignant tumor is cancerous. They lose their anchorage dependency and can leave the tumor site. When they do, we call it metastasis. So if you ever hear of someone saying, oh, they have metastasized cancer, this is what they mean, is that a malignant tumor spread somewhere else. Oh, there it is right there. When a cell separates from the tumor and spreads somewhere else in the body. Unfortunately, cancer can affect everyone. That's it. Life's about risk management. However, there are various things that you can do to minimize your development of cancer. Don't smoke anything. Cigarettes, vapes, anytime you're combusting something into smoke and inhaling it into your lungs, it's a carcinogen. Nicotine causes a mutation in cells at high rates. Smoking is like the worst thing you can do for your body. Eat healthy, drink water. Fatty foods and dehydration can affect the function. Protect your skin by using SPF. Use sunscreen. It does not matter how much or how little melanin you have in your skin. The sun doesn't care. What, you think you're tougher than the sun? Put on some SPF. Sun is damaging to skin cells. It could cause mutations to occur after exposure, and that has to do with UV radiation from the sun. Put on sunscreen. The tougher than the sun line I got from Mr. Urban. What do you think? You're tougher than the sun? If possible, ask your family of any history of cancer, and get regular cancer screenings. Cancer is sometimes genetic, so you can have a genetic component to it. Doesn't necessarily mean it's inevitable. If your mom had cancer, it's not inevitable that you're going to have cancer, but it may increase risk factors, right? Lead to higher chances of. Great. That's it. Thank you for joining. I'll see you in class.